Good morning, everyone. To those who are in the lecture room and to those who are online, thank you for joining us for the talk, Decoding the Human Brain, a Neurosurgeon's Experience by Dr. Pascal Zinn from University of Pittsburgh Medical School of Medicine. He's a director there. The bio is in the meeting invitation, so I'm not going to reread it. And without further ado, Pascal, you have the floor. Actually, you can.
Yeah, I have a microphone, if that works. Okay, thank you so much for the invitation, Ivan. It's a real pleasure of being here. I hope everybody can hear me. I really love this to be interactive. I think if we just, everybody can chime in from either, here or from online. Just please interrupt me and we'll get through this together in the next hour or so. Decoding of the human brain, I know probably you guys understand much more about this than I, but I tried to catch you guys a little bit with the title. But I want to show you really my side of what I do. And it is a lot about, what I do is kind of a dirty business. There is, blood, there is patient brain, there is open skulls, and we try to create that clean interface with computer to maximize tumor resections and really improve patient quality of life and life expectancy. But this is really another aspect that I believe probably you guys don't see every day, but it's very, very related to what you guys are doing. So I'm super, super excited to be being here today. This is literally a huge team effort, and it's not just neurosurgery, it's neuropsychology, it's electrophysiology, it's neuropathology, neuro-oncology, radiation oncology. You can read it there. But also neuro-nursing, neuro-rehab, and our trainees, residents, fellow students, that's extremely important. And we study the scientific aspects in my lab, but there is countless collaborators, and there is 2 or 3 really important collaborators. One is Natalie Sherry. I'm going to show some of her slides. She's our neuropsychologist that is there during the surgeries, during the awake surgeries. And then Philippe Leduc and Raoul Panat are from Carnegie Mellon University. We're developing tech with those together that we try to get clinically approved and translate on that. I really don't have any major disclosures, but the intraoperative pictures and videos can be a little bit graphic, so I hope you can take it. And then I use ChatGPT for some illustrations, as you will recognize. I love that. The brain is extremely, I would call it a complex electrochemical construct. There is 100 billion neurons. A single neuron can interact with 15 to 20,000 other neurons. A single neuron can have up to 100,000 synapses. That's on the higher end. There can also be as little as kind of 1 synapse, right? But some of you may have already done the math. That's a quadrillion of connections that you can form. That's like a 10 to the power of 15. That's essentially probably about a 1/6 or a 1/7 of the proposed atoms in the universe. It's kind of mind-boggling if you think about that. I looked that up yesterday. So that the largest computer chip that was ever built with transistors on one chip has 2.6 trillion chips. I'm sure it's much higher by now, or you guys know better, but that's not all that different than the 100 billion of neurons, but essentially a transistor is just an on and off switch. A neuron is a infinitely more complex entity, right? So man versus machine. I like to play chess, but chess is unsolved today with our current computer power. There's about 10 to the 40 amount of legal moves in chess. If you go to illegal moves, it's about 10 to over 120, I think, the power. So like more moves in chess than atoms in the universe. So that's why Deep Blue, at that famous event in 1997, when Deep Blue beat Kasparov, Deep Blue at the time was able to perform about 200 million or anticipate 200 million moves per second, which when you think about the amount of total moves that are possible, this is absolutely nothing, right? Even the most complex, most powerful computers nowadays can only about anticipate 10 to 15 moves in chess. Because as you understand when you look at that number of the possible moves. But interestingly, modern chess engines calculate about 50 to 100 million moves, so less than Deep Blue even, and that only at the absolute maximum hardware resources. So even your iPhone can essentially be used now to be cheating at chess, which happened recently, I think. And these new models are pretty powerful. Anybody has any ideas why that is, or any thoughts on that? Please just chime in. Let's make this a little bit interactive. Online? Yeah.
Maybe it can prune the search space more effectively early, and then it doesn't have to do that many moves.
You know, I don't know the details of it, but certainly like novel models, like neural networks, that is being, used, and some form of informed decision making. Stored games, I would say, that are just more readily and more smartly recalled, and then, the vast amount of moves per second that are predicted are just optimized by additional input. So I think the software has gotten much better. Not, the calculation power is not what defines, I think, how good, a model is necessarily. And we'll get back to that, but please keep that in mind, kind of this man versus machine thinking throughout the talk, and we'll come back to that. And please just interrupt me, raise your hand, or chime in from online. Going back to what I do for a moment, I work at the University of Pittsburgh, at the University of Pittsburgh Medical Center, and DALL-E ChatGPT is depicting this beautifully here. It's a pretty large effort. There is locations all over Pennsylvania and internationally in Italy, Ireland, China, Croatia, Kazakhstan. It's a 28 billion revenue like institution. But so when I made this slide, I was thinking maybe I shouldn't be bragging here at Microsoft about any large institution that we have. But I think for the medical environment, this is still pretty large. We own about 45 hospitals, 9,000 licensed beds, 800 doctors' offices, more than 6,000 physicians. And in fact, not many people know that we have one of the largest, I think probably the largest neurosurgery program in the country by volume, with over 12 to 13,000 cases. And the largest training program in the country in terms of raising our residents that takes 7 years to go through neurosurgery. What I'm leading is the neurosurgical oncology program, where we deal with brain tumors, with brain metastasis, cancer that arises in the body and travels to the brain. We also deal with spine tumors, metastatic and primary spine tumors. And we perform the focused radiation ourselves with our radiation oncology colleagues. We work on therapies in the lab. We publish papers. We train the next generation. We apply for research funding. We try to innovate. And I'm also leading the biobanking effort, where we bank every single specimen from every patient and then genomically profile those, and building essentially a huge database with thousands of patients at this point, with all annotation, interestingly, MRI analysis, which are, we use AI paradigms to analyze that, and all the genetic information. You can imagine how big that data set is. And it's invaluable in the age of AI, how to leverage that to predict about anything, right? AI is only as good as the data input, I believe, but you guys would know better, certainly. So what I do, what I really care about is brain tumor patient care. And in my program, I really am doing this very patient-centered. I want to call this personalized medicine. Every patient gets their personal treatment, because no patient is the same and no brain tumor is the same or in the exact same location. And thinking about what I mentioned about how complex the brain is, you can think that those pathways can be interrupted at any given spot in the brain, can can really surface, the oddest syndromes. There's beautiful case stories about the human brain by Oliver Sacks and Antonio Damasio and the likes that I would recommend to read. We also, which is quite new, we perform neuropsychological assessments here with my colleague, Dr. Natalie Sherry, pre-op, intra-op, and post-op. So we have very detailed, in-depth neuropsychological batteries for every patient. And what has really changed to when I trained a couple of years ago, this is now all quantitative, and we can analyze it, we can do math and stats on that. Surgical safety optimization, we have a center for perioperative care. Every patient goes through, because we not just care about the brain, we care about the bodies, the heart, the kidneys, the lungs, are they good? Anesthesia and cardiology clearance, very important. And then functional imaging, I'm going to show some examples about that. We really care about studying the brain before we open it. What this talk is going to focus on a lot is really the interface of how can we intersect with tech and the brain. And it is about mapping, about monitoring, and about language testing during surgery. Then neuro rehab is very important, of course. Tumor genetics, personalized therapy. A tumor is not just a tumor anymore. It really matters what genes are amplified or deleted, what is driving the actual cancer growth and proliferation of the cancer cells. And all of this is then kind of taken together into a multimodal approach. Any questions, comments so far? So what I really have specialized in over the last, I'd say, 10 years is doing awake brain tumor surgeries to preserve function of the brain. The brain itself doesn't have any pain receptors, so you can essentially just cut it out without causing any pain. You have to anesthetize kind of the skin and the dura mater of the brain. You may remember that movie, Hannibal. Lecter, who kind of eats, the brain of a patient, not a patient, of a victim, and that person doesn't have any pain. I'm sure you guys saw that movie. That's actually like that. We can literally cut out the brain without anesthetizing it, and the patient is just talking to us. We want to maximize extent of tumor resection and decreasing neurological deficits post-op and creating a safe environment for that. It's always a trade-off. Like the patient who is awake has an unprotected airway, and we're going to really have to mitigate. Let's say if the patient has seizures, we're going to have to control that. So it adds a complexity to the case. It adds some risk, but it also really protects speech and cognition. Like everything we do in medicine is literally a trade-off. It's a fine line we walk, and I always explain that to my patients.
So you're operating and the patient is awake and you interact with the patient?
Yeah.
While cutting pieces of the brain?
Yes, yeah.
Wow.
And I'm going to show that. This talk should really serve as showing you, pretty graphically, how this works and how we do that and how we can protect the brain by using that brain-computer interface technology and essentially really decoding the brain, being it on the electrical and on the neuropsychological side at the same time. Now, why do we remove tumors? I'm going to go quickly over the next 3 or 4 slides, but essentially a study in Norway showed that if you just do a biopsy of a specific brain tumor versus if you remove it, in one hospital they actually always removed the tumor, and in another hospital they just took a little sample. And in that hospital where they removed the entire tumor, the patients lived way better and longer. That was a few years ago in 2012. But that's not that long, that's just 12 years ago. And
the biopsy does not remove the tumor?
No, it just diagnoses it. Yeah.
But so it's not treatment in general?
No, but then they may have gone on to get radiation or chemotherapy for it. But that study, wouldn't be possible nowadays anymore to randomize patients because that data is now very solid. But interestingly, that was just a retrospective analysis in a hospital that just had, in the same country, close by, but one hospital just had the practice of just doing biopsies. The other hospital was removing the tumors. So we really know that removing the tumors will prolong lives. And the extent of what we remove also matters. This graph just shows, mark my word, that the more we remove of the tumor, the better we can control the tumor. Now, but the brain is not like an arm where we potentially, God forbid, can cut off the arm and, let's say, cure a cancerous tumor of the bone, potentially. If we do that early on, we really can't, we just can't remove the brain. And the brain, although doesn't carry like function in every aspect that we can measure, in every aspect of the brain, but our resections are always limited by brain function. We want to preserve who we are. We want to preserve, arm and leg function. We want to preserve speech and cognition, and potentially or always try to get patients back to what they were doing. Yeah.
Pascal, what does the brain tumor look like? Are we talking about a single kind of tumor, or are there different kinds of brain-related tumor? Because like for the rest of the body, tumor being removed, I think the body can regenerate that particular part of the body. Like you can repair, but you can't really repair a neuron, right? Once it's taken out.
Essentially, the body can't, if you have to remove part of an organ, except perhaps for the liver that regenerates very rapidly, the other parts of the body really don't regenerate. If you have to, remove half of the stomach or something, then that's just gone. Same in the brain. Neurons don't really regenerate, and if you, let's say you take out the motor cortex, the patient is just paralyzed, and that cannot be recovered. Yeah?
What type of tumors you see? Like, is it usually one solid or multiple? Like, I'm just curious, what's the...
I'm mostly talking about tumors arising from within the brain here that are diffusely growing throughout the brain. A brain metastasis, for example, can be very circumscribed. We kind of remove it all in one. There may be microscopic disease in the vicinity after removal of a brain metastasis, that let's say a lung metastasis that travels up. And then we boost the cavity with radiosurgery, with focused radiation, and have excellent long-term control of that. That's a different story than a tumor that arises within the brain called a glioma, or the worst kind of it, the glioblastoma that, I'm sure everybody has kind of heard at some point about.
So what is the probability in this curve? And what is means N there? Sorry.
Yeah, so this curve just really shows that we have to, this is a Kaplan-Meier curve, basically. It's a survival curve. It shows that when we do a gross total resection, we remove whatever the MRI shows. Patients live the best. But if we cause a substantial neurological deficit at the same time, we really invalidate that benefit of removing even the tumor. Then we better do nothing. And the N means no neurological deficit and neurological deficit. So the worst outcome here, if you look at this, the red curve, is basically causing a neurological deficit, and the extent of resection is kind of lower than 95%. So that's why, just want to make the case that if we remove the entire tumor as seen on MRI, we better don't cause a deficit. So the last couple slides really kind of are a rationale why we do all this. Now, the patient's brain is really complex, and especially the left side or the dominant side carries speech function, and we now understand there is not just two speech centers. This has originally been kind of the Broca's center, and this over here has been the Wernicke center. This is more for like fluent speech production, for understanding of speech, and for making sensical speech. And Broca's area is really just the motor component of speech. And we really understand now, what this does much better, and that it's much more complex. We can't just see this readily on an MRI, even on a functional MRI. We really have to map it out in surgery. And this shows the pathways, the white matter tracks in the brain over here that all play a role with cognition and function. And we nowadays we can visualize those on a diffusion tensor imaging MRI beforehand, and we can study that and correlate it with our neuropsychological assessment to get be as prepared as we can going into the surgery. So left dominant versus right dominant. This is MEG data, magnetoencephalography, that every one of my patients goes through before we do surgery. And then you can really show dominance wherever you see those yellow spots here. These are activated brain regions during different testing, word naming, speaking, and then I want to know on which side I operate, if that side is dominant or not. And I'm more so inclined to do the surgery while the patient is awake. If we're talking about the dominant or speech eloquent, we also say brain region. So you appreciate now that we do a lot of preoperative testing of all sorts of modalities to understand, or just to be well prepared before we go into a brain tumor surgery. On the right here, you see, again, a high-definition fiber tracking protocol that we developed at the University of Pittsburgh that is very, very high resolution, and we can zoom in on every specific track, basically, that I showed you on an illustration in the slide prior. And this can be combined, of course, with the MEG data also, and then put together as a whole. And when we sit in clinic with the patient before surgery, we go through all those pictures and just show them, and it's very obvious for them to understand.
MEG is magnetoencephalography, right?
Yeah.
What is the principle of HDFT?
HDFT is diffusion tensor imaging, which essentially looks at the vector of water molecules in the brain. So wherever you have tracts, the water molecules are aligned along the tract, and they diffuse along the tract, as opposed to molecules outside that. And it's sort of an arterial spin labeling. You kind of activate the magnetic field, then you relax it, and the molecules kind of switch back. They're dipoles.
So it's built on top of MEG?
It's just one of the MRI protocols. We can get it routinely, and then this is just a special post-processing that we use here for this very high resolution. This is ChatGPT's take on neuropsychology. I found that very interesting. In fact, I'm just so excited showing you all those ChatGPT generated pictures. Because it tells us a lot on a meta level, it tells us a lot about what's going on in the world, and that's why I put that in. And please by no means take that as actual factual data, but it's just a testimony of a.
You're quite good at getting the model to generate high quality images. I'm never this good. Like
Nice processing, yeah.
This is some master level content.
I made it a rule when I did this. I just asked it once. So that's my first try for all those images. It's remarkable, and it really lets us see how it thinks, which is worrisome and exciting at the same time, of course.
So it is more the general perception of that word, how visually it is, what the average person thinks about that.
Yeah. And the neuropsychologist here is a woman. Like you can read a lot into this, and I would have predicted that because it just makes assumptions, right? It generalizes it. I could give a whole other lecture on that. So what Dr. Sherry, she is a woman, does for us, and we have a joint lab meeting actually together. We have about 5 people now working for both of us, and this is a joint effort. So this is a busy slide, but the takeaway here is really that historically we just used to, when the residents call me and tell me, oh, we have this new patient, and they're moving all their extremities, they're alert oriented times 3, whatever that, to name, place, and year, and kind of they're walking, right? That has been for the last 50 years, like a neurological exam in neurosurgery has been very simplistic. Now we're really changing things. Dr. Sherry is doing these 2-hour test batteries on every single patient, and we're actually, it says here her report. This is straight out of her report. So here it says there's trouble expressing his thoughts. There is neologism, semantic paraphasic errors, and non-cohesive expression of his thoughts, issue with reading, and so on. This is not just, he's moving all his extremities well. And you would be surprised how simplistic this was until in most places it still is, actually. Like, worldwide, this does not exist yet, but we're just starting to do this. So this patient is demonstrating intact auditory working memory, auditory comprehension, visual spatial judgment, mild impairment in problem solving, visual working memory, moderate to severe impairment in simple sequencing, cognitive flexibility / shifting, processing speed, aspects of language, verbal fluency, object naming, reading, comprehension, writing, and repetition. Like, it goes on and on like that. And we're sitting together then with the team and looking at all the preoperative imaging, the data, be it fMRI, MEG, high definition fiber tracking, transcranial magnetic stimulation, like all of it potentially. And we're sitting together and reviewing approaches to tackling specific tumors.
By the way, ChatGPT has a very good application of converting this free text into answering a specific question as you may have.
Yeah, I'm sure. And then I guarantee that will be an absolutely must-have part of medicine. It already is. It's abundantly used. So different tasks we ask the patient, for example, picture naming alone. I'm not going through everything, but it really tells us a lot about how the brain works. And we do this before and during surgery and then after surgery again. And in fact, the only way you learn about the brain is either if you knock out different brain regions with electrical stimuli or essentially if you cut it out. And a lot of progress has been made during the big wars worldwide when there was so many injuries to the brain. That's when the first case descriptions were published about what brain areas are specifically doing. And when we do language mapping in surgery, we present the stimulus, and while the patient is performing a task, a very specific task designed for that very patient, and then we knock out with an electric stimulus on the brain, we knock that capability out, and then we can, only then we can know that this is the area that we want to preserve. So this is Natalie and her setup during surgery, and the patient essentially has an iPad focusing the patient, and this is somewhat mirrored to what she can control, and then she tests him, and in the background you see my team doing surgery here.
Do you get to draw them?
Draw the pictures?
Yeah.
No, like we have, these are all sort of tested and published, like pictures, and pretty clear sometimes. And the questions can be hard sometimes. While I do surgery, I listen to what she does, and I'm like, okay, that's not easy. But maybe I just focus on my surgery better also. In any case, then there's common errors that she finds, and she's naming those errors while I do surgery. Let's say she can say, this is a speech arrest, this is an error otherwise, this is a circumlocution, like something like if the patient, there's a key there and the patient says, oh, this is something I used to open a door. Like, there's very specific brain areas that just produce errors like that. And it just becomes more clear the brain is mostly like a big wardrobe where you have drawers, right? This drawer has, picture naming. With this drawer has like face recognition, and there are specific diseases then that some patients can't recognize faces. They can see the person, but they just don't see the face. So that's, for example, called prosopagnosia, if you just can't see a face. So it just tells us a lot about the brain. And then there's very specific functions to parietal lobe, frontal lobe, temporal lobe, and then a brain area that I'm very interested in is the insular lobe, which is kind of covered by the frontal and temporal lobes deep in the brain, and has a lot of function in personality, perception of olfactory stimuli, smell, and integration of smell into personality and decision making. Very, very unknown brain area that there's really no precedent in any rodent or any other primate, I would say, about the development of the insula. So a lot of my research focuses on the human insula, but what is incredible that at some point in the future we'll be able to really decode all those brain areas and specifically understand what is stored in terms of memory, perhaps, as we talked before, how memory is consolidated and recalled, and where actually it is stored. If you essentially sleep deprive an individual that has read, let's say, or memorized a few items, they cannot consolidate this into long-term memory, which happens mostly in the cortex or prefrontal cortex, not in the hippocampus, which is a structure known for a short-term memory function. So there's very specific mechanisms in the brain how you consolidate memory, and you can't disrupt those pathways.
Where is the motor and sensory cortex? As far as I know, they are kind of here on top of the...
Yeah.
They're under or in between those two areas?
This is the motor cortex here. It's very back in the frontal lobe, and the sensory cortex is the first kind of gyrus in the parietal lobe.
Does the proximity mean anything for the signal? Or
Of the like motor and sensory cortex? Yeah. It's a good question.
Because I'm thinking much like highways, like just because they're close together, it doesn't mean that you just make this connection. You could just go all the way around, right?
I think it's probably of evolutionary must be of benefit. Most of the things are in the brain, not everything, interestingly, but it's very helpful if those cortices or brain surface areas are in close proximity, because you actually need a lot of feedback, sensory feedbacks to, let's say, you walk, right? If you walk, you have muscle that makes you walk, but I don't have to look at my legs. I know where they are. And why do I know where my legs are? Because I have feedback at all times that actually is coming to the brain. If you remove the sensory cortex in patients, sometimes we do that when we remove tumors. These patients cannot walk anymore right away, but once they look at their legs, they actually, oh, here is my leg, I can walk safely. So sometimes you take away different aspects of bodily function that can be compensated very easily with just maybe, let's say, a visual cue.
Someone has
Yeah, please. Is it John? You have a question?
Yes, thank you. I think I heard you say that during the operation, you're able to suppress an ability and see the effect it has on their ability to complete a task. And I'm curious how much the preoperative imaging plays into your accurate ability to identify what the reaction is going to be. Like, how much better is it with the preoperative imaging versus, let's say, you didn't have it going in?
So things like let's say in a sort of a virgin brain that hasn't been operated on, or that has not recovered, let's say, from a stroke when the person was like a child or something, it's very reliable anatomically to know where motor is and where sensory is. I don't need essentially a preoperative testing to know where the motor cortex is. And I know exactly the homunculus that everybody has seen. It's a very specific area in that motor and sensory cortex for the face, for the tongue, for the thumb is very big, the hand is very big, and so on. So wherever you need fine movements, that's overrepresented in the brain surface, basically functional anatomy. And for that, I don't need that. But what I need in surgery is to stimulate it to find where exactly it is, because it's not always obvious once you have an open brain, a narrow window that you look into. You don't really know which, are you in that, parietal lobe now, or just one gyrus further ahead in the frontal lobe over motor. So for that, we need direct stimulation. But for language, it's very interesting. It's very helpful to know where the dominant, most patients are left dominant, but often it can flip and it can switch, and there can be a co-dominance. We're just scratching the surface of understanding that now, because we've done hundreds of patients doing this. So it's very helpful for language and to assess the localization or co-localization of where the tumor is, or what approach I want to choose to remove the tumor. It's also very variable, again, for language, and it's really much more sure, and the gold standard is direct cortical stimulation. So at this point, we don't have any preoperative test that would replace what I do in surgery. At some point, probably those techniques get much better, and we can perhaps just kind of, with laser ablation or something, kind of treat the tumor, but we're really not there yet.
So you can
Yeah, that's kind of what I was getting at is maybe you're moving towards that, but we're not there yet.
Yeah, we're really not there yet, and I think there will always be, I don't think I'll be jobless anytime soon, but and there's always those tumors that are just very, very large, and the patients kind of present obtunded and have so much mass effect that it's an immediate life-saving surgery, and no medical or molecular therapy will ever be able to treat that. So I think there's always going to be a benefit of having a surgeon actually that can open in the near future, but hopefully we'll be able to treat that otherwise at some point. Yeah.
Go ahead, go ahead.
So you can see with your bare eyes the region that responds to the stimulation?
Yeah.
Like, does it look differently? Does it move?
No, you cannot recognize it with your own eyes. You can see the surface of the brain and you can stimulate it, and I'm going to show you that. And but you don't. It doesn't look different. That would be really interesting if, like, oh, this patient is like thinking about the car or about a pot of flowers, and it doesn't show up. It's really those hundreds of billions of synapses just create that, and we just don't understand why. It's really not just an on-off semiconductors thing. It's like it's impossible to understand, I think.
But you hold a scalpel
Yeah.
during surgery
Yeah.
and do the cut by hand.
Yeah.
Wow.
It's all done by hand. So how do we do this awake surgery? We're, as I touched on, we're trying to understand pre-op, leading up to the surgery, what tumor, where is the tumor, and what is involved with it, and what are the tracks that we want to preserve. And the technique is, while the patient is awake, we have this so-called Ogerman stimulator. Jeff Ogerman, I think, was for large parts of his career in Seattle, actually. And he designed this probe, and we use a slightly different approach nowadays, but this is essentially what we're doing. It's a bipolar stimulation of the brain surface, and we're knocking out that function while our neuropsychologist testing the brain.
So,
Yeah.
can you go back? So those are 1 millisecond pulses with frequency of 50, 60 hertz.
Yeah. We can adjust it and play with it, and then I kind of go up to maybe 12, sometimes 14 milliamps of stimulation intensity current, depending on, that requires a lot of experience because we don't want to cause any seizures, and the more amplitude you use, the higher likelihood is that you cause a seizure. But this is, that's where just, I think experience is really, really important doing that to make it safe, and you just got to have the repetitions like in anything. And we do now probably about one of those very complex awake surgeries a week, which is a relatively high, I would say very high volume for any one surgeon. And so looking at an example here of a brain tumor, these are 3 projections. This is called axial, coronal, and a sagittal view. This is obviously a side view here. You see the eye is a little bit cut here. And this is mainly in the temporal lobe here. This is the temporal lobe, which is expanded and very inflamed here. You see that? And it pushes the frontal lobe up. And this is classic location for speech. It's left superior temporal gyrus, meaning high up in the temporal gyrus. This is, in general, something that a patient presents with profound speech deficits, also caused by all this inflammation. And then the tumor is not just that ring of enhancement on the MRI, it really goes beyond. We nowadays use dyes that light up the tumor cells, and under the microscope with a fluorescent filter we can visualize that dye and can go way beyond even the MRI. That's why we don't look at real margins in the brain, we just look at, because we cannot just clean out everything at all times. It's really, we have to identify anatomical borders and then functional borders by stimulation and kind of really understanding where is the limitation of what we can do and how much do we have to preserve so the patient actually overall has a benefit, right? If you get the entire tumor out, but the spouse doesn't recognize the patient anymore, then we have a problem. We haven't done any good. Yeah?
Well, a couple questions, but it might be silly ones. I'm just curious. What makes this ring like lighter, and why is it like emphasized? The other thing is, does the structure or the geometry of this change from one language to another language speaker, or beyond that, multi-language speakers?
That there is profound changes in brains of bilingual, trilingual people. That's very well described. It wouldn't really, have a correlation with where the tumor is and how the tumor spreads. These are tumors that hijack the white matter tracts of the brain, and they just spread out along those tracks for the most part. They can also cross like significant borders in the brain, rarely if they're much more aggressive. But what I'm doing, I'm chasing those tumors across those white matter tracts that connect different brain areas.
So the tumor here is the highlighted part.
Yeah, the tumor here is all this. Mm-hmm. And inside it was growing so fast that it's devitalized tissue. It starts as a little bright dot, taking up the contrast we give to the patient. And that's why we can only visualize it. And then it just grows exponentially, right? Cell division out into the periphery. And inside it just dies the tissue. So the active tumor is the ring, and that rapidly kind of spreads out. But the tumor is not just the ring, it really goes beyond that, and cells are spreading out. There's a scarcity of oxygen and nutrients, of course, here, and there's a lot of research about that. The cells don't like that, so they're rapidly migrating towards areas with more oxygen and nutrients, which is into the periphery. That's why we can never fully remove it. There's always microscopic disease left behind. But the data also shows the more disease we remove, the better we can treat the residual disease. Unless we cause any deficits, that kind of really invalidates what we do. That's why it's so important. I want to say that's why my job exists, I guess. Now, how can we remove that? So, the tumor is exposed here. You see the brain underneath. You see how the brain pulsates here. And that's just very normal. That's the heartbeat. That's the patient's breathing. That all impacts on how the brain looks. And you see underneath here is really the brain tumor. And the surface looks relatively normal. You can recognize something is not right here. Look at the configuration of the blood vessels and so on. You see the electrodes, the ground, and then every single time we have electrodes on the brain. And we, in fact, study brain anatomy with it. We can identify the motor and sensory strips just with stimulating and recording. And then we can stimulate continuously throughout the surgery and then record from arms and legs to look for integrity of all the motor pathways. And we look at all the integrity of the sensory pathways by stimulating peripheral nerves. So at all times we know those pathways are intact and not disrupted.
So you can have real-time monitoring
Yeah.
of the stimulation.
It's all real time, like every single patient is, really lined up with electrodes, and we're stimulating and recording non-stop. Even if they're asleep, you can do that.
What is wrong with that region again? Because you said like
It's expanded here. You have to take my word for it, but this is just, it's swollen, it's kind of protruding at us once you open the skull.
Okay.
So you have constantly ECoG electrodes, which we saw in Zinn, and constantly stimulate the legs and the arms and get the...
And the brain, stimulate the brain.
surgery.
So I basically stimulate and record at all times from the brain and from the peripheral nerves.
Wow.
And you measure all that connection, and that can be pretty long, right? So it's just a miracle. Like there is, 6 feet of wiring going through our body, and we can just in a millisecond assess the integrity of it and correlate it with if the patient can walk after surgery or not.
What about a neuropsychology test? Is that a simulation or that's an observation?
That's an observation, but during that we're also recording all the brain waves. So in fact, there's a lot of advanced, predictive models we can run about what brain activities are active with different tasks. And that gives us an unprecedented view into, this is not just a helmet of electrodes that you put on with, there's a lot of impedances and resistance throughout the scalp, hair. This is like pristine signals directly from the neurons recorded. All has pros and cons, but close your eyes if you can't see this, please. But this is an
Up from 1. Just start at 1 and keep going. 1.
This is an exposed brain. Just wanted to stop this for a second, but an exposed brain, and you hear Natalie Sherry, my neuropsychologist, in the background, and we're getting a baseline testing here, and then we're actually using that stimulator.
That is incorrect. Semantic paraphagia.
So she gives me an immediate feedback
That is correct.
of what the patient is doing on the other side, while the brain is exposed and open, and I stimulate.
Good.
And then can mark that brain surface with what its function is.
So this is the surgeon's hand.
Yeah, this
and
is.
what we hear is basic interaction with the patient.
Yeah. And
Is it just for data collection purpose, or does it actually, is it part of like the treatment for doing that?
yeah, you want to really preserve that area. If that area carries function, I'm not going to remove it.
Oh, so if you hear the beep, you just don't want to touch there. It's basically, okay, I see.
Yeah, not the beep, but if you. So I wanted to show this movie here. Oh, here. Okay.
Now,
So here you see both sides.
what is this now?
Alright.
What is this now?
Alright.
What is this now?
Oh, okay. That's here from.
So there you saw, like when I stimulated, patient had a picture of a fly or something and just couldn't name it. So that was really a speech arrest, and this is an area we're not going to remove. And only that really allows us to maximize resection and preserve function. I think I showed that already. And then here you see while I'm working. This was while I'm working, and here you see zoomed in on the iPad that tests the patient at the same time while I continue to work and remove the tumor.
Good.
And while this is going on, we monitor the entire motor function and sensory function as well while the patient is awake. And it's very reliable.
Perfect. Very good.
So this is kind of a summary slide for some of what I mentioned. You present the stimulus to the patient while they're awake, and we have electrode strips on the brain. Literally any site is accessible because we can slide those grids in, and we collect that signal. Everything is stored from all the different grids, and it is time-stamped with what we do on the iPads. And we can literally, we have a study now running where we present an olfactory stimulus, like different smells, and we record from different sides of the brain. And we're stimulating the brain to see how we can interrupt those stimuli and perception and sense of self of the brain with insular stimulation of that green area that you can appreciate that there in the middle. So you can think about the amount of data we can collect and the amount of ultimately understanding of brain waves and its implications on patient behavior and processing. And as you may think now, like the advent of advanced computer algorithms, as I'm sure all you are working on, can then hopefully help us make sense of that data and really understand the human brain. We don't understand anything yet.
I think there's a question.
Yeah?
A question?
Yeah?
Yeah, so you're doing these stimuli at the surface, but then you're cutting in. I mean, the brain is three-dimensional. So as you go deeper, are you also doing stimulus down deeper? I mean, as you're cutting down, you've also got to worry about, well, what's underneath, or do you have a good model to know that, oh, if I see this thing or don't see this thing, I can cut down so far. Like, so how do you know how far you can go down, and how can you put probes like inside or only on the surface? I mean, there's an exposed area, so I guess that's what's accessible to you.
Yeah, that's a great question. Yeah, the surface is very important, the cortex, because if we take a small cortical part, that we call that the somatotopy. There's a very organized cortical surface. If we remove a small area, that function is gone, potentially. Now, deep in the brain, there is white matter tracks connecting those areas that matter, and we don't want to take those either. So we stimulate deep the white matter tracks, and then our neuropsychologist just continues testing. And whenever deep in the brain, it's less kind of an all or nothing phenomenon. Somehow there is, I think, redundancy in the white matter path. Mm-hmm. And when we approach an area that carries important function, we'll see that right away, and then I can redirect my approach or my tumor removal direction. And then that's a continuous feedback, like a monitoring, neuropsychological monitoring while I do the surgery. And then I stimulate to kind of anticipate. And usually there is, we have different, we call it subcortical stimulation, not surface, but deep. And I can gauge very, very effectively how far away in the tissue, in millimeters, depending on how much milliamps I use. So I can gauge like, oh, those tracks that I'm disrupting now are perhaps a centimeter away, or even more, or much less. And then it's really the matter how you push the limits in terms of how close do you want to go. And that differs really from patient to patient, from tumor to tumor, how edematous, how inflamed is the tissue, how spread apart are the fibers that matter, and how much tumor is infiltrated there, as seen by that dye that lights up the tumor cells. And then every single time, I think the most difficult decision making in my job is really how much do you take of the tumor at preserving function, and really, even more important is when to stop, I think. Did that answer?
So
yeah, that's great. Thank you.
Stimulating is basically marking the territory of your cutoff region.
Yeah. And
Very good. What color is the sky?
Red.
What color is the grass?
Red.
What is 1 plus 2?
Red.
Very good.
so you see, there's simple questions, but she tests my patients kind of rapid fire based on pre-op baseline that she knows, and she knows exactly what those patients can do and cannot do, and based on that we tailor what paradigms we use during surgery. I think you had a question.
Sorry, if I missed that, how long on average each surgery takes? I know it depends on the tumor, but
Yeah.
some kind of ballpark.
I'd say between 3 and 5 hours or so. I try to be quite efficient. Sometimes it takes an hour longer because the patient doesn't wake up, because I put them to sleep for opening, because that would be quite traumatic if you hear, being drilled in your head and things like that. So I put them to sleep, then we wake them up, and that takes anything from like 15 minutes to maybe an hour till they're awake, and we have a good baseline so we can start testing.
So basically, and you collect data that you described for all those three, 4, 5 hours.
Yeah, everything is stored and time stamped with everything. Yeah. And interestingly, we're just now starting to tap into this. We have multiple grant submissions and approved research protocols now, where we interact into neural circuits with stimulation, and we're recording everything. And so.
There's a question on that.
So I have a question.
Yeah.
So when you're operating, have you pre-mapped the area of the tumor by correlating with the imagery, or you have some real-time mapping also on the tumor?
So it's quite high tech. So we have an operating microscope that actually, if you look through the microscope, you have projected the preoperative imaging, the MRI, the functional imaging, all the tracks are projected large on monitors wherever I focus with the microscope. And this is kind of almost like kind of a ceiling mounted large microscope with very high resolution with 2 eyepieces that my assistant can look through. And it's really augmented with all the data we want.
Technically, this is augmented video, augmented reality to assist the surgery.
Yeah. And in real time, wherever you look with the microscope, you kind of see on the monitor exactly the slices you are with the tracks highlighted. Now, I have a lot of ideas how to improve that, and it would be nice to actually use HoloLens or something to implement that. That would be a game changer, because the microscope can be also cumbersome and so on. And it's just doing this every day, kind of, you realize how antiquated this is, in fact. And I'm excited for the future, how this is all going to improve, actually. Yeah?
I have a kind of a dumb question, but like, so those microscopes, how do you manage to see this with your glasses on?
Essentially, it's really just a binocular. And you could take your glasses off. I don't. And it has eyepieces that are adjusted, and it's zero to your kind of visual capacity, but you could adjust it to even without glasses. But.
Thank you.
We also do have goggles we wear, right? These are head-mounted goggle systems that have different augmentations, I would say, with blue light for different fluorescence light and so on. But it's cumbersome. You take off your kind of loop goggles, and then you go under the microscope. Then you want to put your loop goggles back on again, and so there's so much room for improvement there, and I'm baffled why it hasn't been done. So again, a little more data here. A motorcycle here. Excellent. And I think you get the idea. This patient, we got an excellent tumor removal here. You can see that.
The lower is post-op.
Yeah. This is pre-op. This is the corresponding post-op, where you can see the tumor is just removed. And it's not always possible to remove all the contrast enhancement, but in this case we were able to, and even a little bit more beyond, perhaps, as indicated by that dye we use.
Pascal,
Yeah.
I'm not sure what the most healthy brain MRI looks like, but there are some crevices there. I'm curious, does that indicate anything, like apart from the operation area, say on like far right down?
Far right here?
Yeah, like on the, is it frontal?
This,
like,
yeah, okay.
Is that normal?
This is a sunken frontal lobe. This is air. So air shows a little blooming artifact on the MRI. It's just, sunken brain, loss of spinal fluid, and that just fills up with spinal fluid. The brain usually re-expands there. But whenever you remove brain matter in a confined space, which the skull is, you have kind of air and kind of water or brain fluid that fills up that space. And rarely that becomes a problem, that kind of that fluid is, or that space just enlarges, and then you sometimes have to very rarely go back and drain that. But yeah, this is very normal. We see that. So I wanted to go back to this. We discussed it already a little bit. But mainly, I think that man versus machine thinking is very, very antiquated and old at this point. I'm thinking it rather should move man to a paradigm that uses man and machine. And when you look at brain, and that's not me, that's ChatGPT. The brain uses 20 watts of energy, which is really nothing. And I'm not an electrical engineer, but again, if ChatGPT and Google is correct, ringing the doorbell is an equivalent of about 2 to 4 watts. So if you see children or your child like, ringing the doorbell like 10 times, that's kind of more energy than the brain uses. Now think about that. What we can produce with our brains and a simple doorbell uses more energy if you hit the button a couple times. If you vacuum at home, this is way more than what the brain uses. And a light bulb, as we know, is like somewhere between 40 and 60 watts or something. Or if it's a super energy saver light bulb, it's equivalent to the human brain energy, about 25 watts.
Our entire bodies consume around 150 watts.
Yeah.
To function.
Yeah. The brain is about a fifth or a sixth of the entire body function. And this is roughly an equivalent, I guess. You can't neglect that, of course, it's calories we use. We produce energy by metabolizing glucose and oxygen. And it's a completely different type of, much more goes into this. It's a little simplistic, I would say. But, modern AI chips apparently use 1,500 watts, just one chip. And that's substantially more. Now, I know you guys are experts, world leaders on data centers. Now, if you take, thousands or 100,000s of those chips, all of a sudden this is an exponential energy consumption that is potentially uncontrollable. So man and machine are profoundly different, I would say, if we give this some thought in architecture, of course, and processing styles. And, any input here is welcome, but I think ultimately there is not enough power on the planet to keep up with our processing demand if you look at this. So there's novel solutions that are needed. Any questions or input? Here I'm getting out of my comfort zone. This is you guys.
I think this is actually a very active area of research as to why we need to address things differently. We cannot go like last couple of years. The trend has been just scale is what you need. Basically, just keep scaling up and up and up. Because it's not driven by science, but like competition, the market. You need to beat the next guy, kind of thing. So, but there's an ongoing effort to make it more like if the brain does it in a very efficient way, we need to do it very efficiently too. But I think the part of the intelligence also comes from that, like there's a economy of the nature that you have to do within all of that computer and budget. So brains, I don't know, maybe you can chime in on that, but the intelligence had to develop because you have to do more with less, because you have that budget in the first place. So you had to learn to generalize. So if you have the endless compute, I don't know if that will bring out the intelligent behavior.
Like the chess paradigm showed that, Deep Blue was very powerful, the size of a car. But every iPhone nowadays with those stockfish engines, beats, the best chess players. So you have to do it probably more economical, and it's a combination of ramping up power and dimension, certainly. That's the easy way, I want to say. Still difficult, I'm sure. But there's something else that we're missing, I think, that will get us to the next stage. I don't know about quantum computers and things like that, but I don't think that will be the answer either.
The brain is a parallel processing machine.
Yeah.
Everything happens in parallel, while all of the modern computers are sequential.
It's really, there's...
Parallelism is minimal. Even the GPUs, they have 3,000 processors. That's 3,000 things happen in parallel.
Like the first computer chips based on vacuum tubes are just electricity or no electricity. Ultimately, that's zeros and ones like you have displayed in front of the auditorium here. So I think power is something very interesting, and I wonder if there is a power problem. So now how could we solve this? And people are trying to solve it. So first of all, I don't think it's man versus machine. It's really man and machine. And I'm going to finish up the talk by asking a question. What if we could harness the power of the human brain, microchips and software or AI all together in essentially man and machine as one? And I think that's where the field is moving to, and there's many efforts now worldwide being explored And this is extremely, extremely exciting. All ChatGPT generated. I love this one. So, and I'm really kind of trying to wrap up the talk here, but I'm just going to leave you with a few more slides. There's several invasive, minimally invasive, and non-invasive options currently available, as everybody's aware. It's really a new gold rush, a race across many startup companies also. And it's just not sustainable at this point. There's no sustainable long-term data now for any brain-computer interface. They all kind of fail. And wetware computer chips and brain organoid, human brain organoid research. I've published some papers on this from my lab, where we did some mechano-compression experiments of brain stem cell-based generated brain organoids, and looking at neuronal function, so we can hook those up to a computer. And then there is several companies and research labs that work on wetware computers, which are still in its infancy, basically. And something that my group has worked on is an extracorporeal brain perfusion system. I'm just going to show 2 slides on that. I don't want to go into too much details. Essentially, when we remove large areas from the human brain that contain tumors, we're able to maintain those samples in the lab now. And it's really not science fiction, but there is some sustained single unit recording possible in the electrocorticography from those specimens that we maintain in the lab. There's sustained immune response, cytoarchitecture, metabolomics, gene expression profiles that all show that the perfused samples are relatively robust, active neuronal signals. And then this is really, at some point, makes us think about brain-computer interface testing, AI, wetware computing, and towards solving the energy problem. Much more on this in the next few months and years when we publish this. Take-home points here. What I've shown you, I hope I was able to give you an insight into what I do, what you have not been exposed to before, and how individualized the awake mapping is actually, based on preoperative tests, as I've shown you, multimodality tests, including neuropsychology, which I think is very, very important. And I'm excited actually to analyzing in a quantitative fashion all that data corresponding to brain activity. Brain surgery leverages nowadays already BCI, and it's innate to what we do to maximize safe tumor removal. The brain is a very complex, I call it electrochemical entity with vast powers. And those powers are maintained at very low costs. And that is just mind-boggling if we compare it with the tech. That has, with a brute force, with exponentially more energy, just in some aspects gets close to what we do, in some aspects beats us certainly. And that really, historically this has been the man versus machine question. However, I think in this day and age, when we're trying to scale up our computing power exponentially, we need approaches that solve that problem with, be it with BCI or wetware computing. And BCI and AI definitely will define the coming decades and beyond. And it's not man versus machine, but really, I think one of the main messages and my vision is really that man-machine as one. I think I'm tremendously excited about that. This is from a awake brain tumor surgery where one of my patients played the guitar. He played professional musician, played in a band. He said, I'd rather, not speak, but I don't want to lose my, guitar capabilities. And that's what we delivered on for this patient. I'm going to close here with, again, the same slide, that this is a huge effort across many disciplines. And I just want to say sometimes I just watch the team, how they perform, and I feel really proud and honored what we have been able to create and kind of, in a way, a little bit conducting the orchestra, sort of. Thank you very much for listening, for all the questions, and we have a few more minutes. I think you can just ask more questions. Thank you.
Question from the audience, questions online? Or have a question online? Go ahead, John.
Yes, so thank you for the presentation. Definitely very fascinating and encouraging. I would love to read more about kind of state of the art. You mentioned doing the preoperative imaging and being able to overlay that with, like real-time microscope view. Is any of that documented? We can learn what the state of the art is and where it's trying to go.
There is, I would say, countless articles published on that, and I think even at my institution, a group uses HoloLens actually to look for AI and augmentation of medical imaging. I think they use it in spine surgery. I think it's, even with your technology, widely used worldwide, and there's many research efforts going into that. But I think the companies that provide those, very old technologies, such as a microscope that is pretty sophisticated in itself, but I think it's just they haven't really realized readily how to adopt all the tech that is already, again, old in a way. So it's like they have a good product, but they're like the cab drivers kind of slept through the Uber, generation and kind of lost business through that. And some of those companies are just not keeping up with it, but we have no other options as surgeons, and we just keep using that. So I think that has to be revolutionized and sped up.
More questions?
What are some of the efforts for rehabilitating the tumor area rather than taking it out?
That really doesn't regrow. Whatever we take out is gone.
No, I meant is there a way to kind of avoid the by rehabilitation? I didn't mean post-op. I just meant are there alternative solutions to tumor removal than, I don't know, using chemical treatment or something?
There is some tumors we treat just with radiation, or be it very radiosensitive tumors, but that's kind of relatively rare. I'm talking about these primary brain tumors. Surgery is still by far the best and most important treatment approach. And then rehabilitation post-op is essential, because essentially, the brain is not like a computer chip. So the primary cortex function might be gone or subcortical function of some subtle personality trait perhaps, but the brain is so still plastic without even generating new neurons. Through rehabilitation and time, you can actually, I think, just by the mere adaptability of the brain, synaptic pruning, a lot of deficits can be overcome. And if that specific deficit cannot be overcome or cannot be kind of remedied, the brain will find alternate pathways to kind of develop, and it'll kind of diminish how the patient perceives that deficit. All of a sudden, they don't talk about it anymore.
That was actually my question. Neuroplasticity at some point became a super fashionable and subject for a lot of talking. To what extent the brain has capabilities to rewire and to compensate the functionality of those areas of the cortex which have been removed?
So it's a very interesting question, and it really surprises me again and again that there is, for the motor control, something that is essential to survival, that you can walk, let's say, or move your, if you can't use your dominant hand, you're severely limited in what you can do, right? That the world is designed to be used with two hands, right, and two legs. There is just one specific brain area that if that is gone, you just can't move your left or right arm, and there's no redundancy there. Think about that. Why is it designed like that? I'm not getting into that too much, but there is some inherent limitations, and then there is functions like memory that are relatively strongly on both sides, and you can quite easily remove one hippocampus. Often even a dominant hippocampus is removable. It just tends to take longer in rehabilitation, but if you were to take both hippocampi out, then at that point you really can't form any new memories anymore, like in that movie Memento, if you remember. But the brain is in many ways very redundant, and then the patient recovers very quickly, and then there is some absolutely non-redundant sites that can be devastating if injured.
More questions from the audience?
I do, but I think there are some online questions.
Online questions? Hey, feel free to unmute and ask the question. No, Tracy, can you ask your question? So the question is there a book in the offering to talk about some of this? Maybe more popular books eventually.
I may write one.
Yep.
I don't know any book that in that detail, speaks about that. I think this is also very kind of, old knowledge combined with some of very new knowledge, what I was talking about today. It is widely published by many groups, of course, but just the aspects of it. But I think it would be very interesting to look at this at the more comprehensive level. I can think of a few BCI books potentially, but nothing that describes it in this depth.
If you give me a list, I can basically propagate it to the chart in this talk. More questions? I have one last one.
Yeah, sure, yeah.
So, you mentioned that one neuron interacts with up to 100,000 other neurons. Neuron itself has one dendrite and up to 10 axons. How it can interact with so many neurons?
Yeah,
So
mostly one axon and then many dendrites.
Yeah.
They're tightly packed. There is I think probably about 20,000, 50,000 neurons on 1 square millimeter or something. And axons can be very long. So it can actually, it's just essentially, it's like fungus growing in the ground, right? That there's entire forests that are connected with the same fungus underneath. That's how the brain works.
So if a neuron gets excited, next neighboring neurons can sense that without being directly connected.
There's a very high chance that there is some form of direct or indirect connection.
Okay.
And then based on, it's a whole neural network. That's why we call it, I guess, the mathematical, equivalent neural network. But it learns as it goes, right? It experiences, it has, kind of successes and defeats the brain at that neural network, and it prunes its connections, what works. That's why, learning how to ride a bike is, you cannot read about it, right? Your parents didn't give you a book or tell you step number 1 is, getting on the bike, step number 2 is like pedaling. That would be useless to you. But your neural system has to experience how to ride a bike. You have to fall a couple times probably, and have to know that you have to pick up speed, otherwise you're not stable and so on. It's a trial and error system, I think. And that's what you see over a lifetime, right? But it's unlimited capabilities and possibilities, right? A human being can learn about anything and subspecialize into anything. And now we're not just our own brain, we're connected by so many modern means of tech. All the human beings that take advantage of that tech are connected, and together we're creating something tremendous, right?
I've read about deep brain stimulation for people with Parkinson, where they put wires in the brain and they stimulate it as a treatment. Do people really walk around with a wire in the brain?
Yeah, all the time, yeah, everywhere.
So they have wire exposed in the head?
It's all under the skin. You implant it, it's under the skin. It goes under the skin to your chest area with batteries.
Oh, so it's continuously stimulated?
Yeah, that there is many bionic people out there. There is closed loop systems to mitigate seizures, like a pacemaker for the heart. That there is deep brain stimulation, there is nerve stimulators, there is spinal cord stimulators. We're entering the age of bionic people.
Because I can never imagine how people have a USB.
Oh, you actually don't say that. Super cool.
There are people with stent actually, which basically the cable goes through the vein to the brain and then next to the heart and where the battery is.
I wanted to, I mean, it's not quite a question, but it's just like, I don't know, like a conversation started. I was curious about your opinion. So one thing that strikes me is that I think for a lot of people, what we call deep learning today is actually quite a surprising fact. Like if you ask a scientist, what's the most impressive thing, you know, he or she will just say, you know, that it works. We don't quite know why it works. But to me at least, it's quite striking that, you know, we provide a few bits of information, be it a word or an image or whatever, and we're trying to modulate this trillion, you know, parameter model of just a few bits, and somehow it works. And I'm wondering, like, from a more neurological perspective, what do you think about this paradigm of like us having extremely highly parameterized models, and maybe the brain does it somehow from a very scarcity of data? Like, what are some of the promising directions for data efficient compute for the brain?
I think I would depend on you guys to tell me that. That's where Microsoft is, you know, tremendously experienced. I think we go back to Deep Blue versus Kasparov here, right? And there's just something about the brain that you do with a gut feeling, and your model is pre-informed by everything that doesn't make any sense. You're not gonna, you know, early on, you know, expose your king in chess and just march across the board, right? And Deep Blue doesn't know that. Deep Blue will consider that move, and then, you know, probably down the road, you know, like a trillion moves down the road, Deep Blue understands that maybe you shouldn't advance your king. But Deep Blue doesn't know that. Stockfish now knows it because that's an informed model, and it also discards those moves up front, saving trillions of like possibilities. So I think it goes towards, you know, making things more economical in terms of having a smart filter there. That ultimately, I think parallel processing, highly massively parallel processing, is the key to making things more simple, and then using brute force computing to where that is actually needed. That's why the symbiosis of man and machine cannot be beat if done elegantly, I would say. Because either way, like if I ask you to multiply, you know, two 10-digit numbers, you probably would take you some time to do. Any computer can do it readily. But if you ask a computer, if you ask Deep Blue to, you know, recognize, you know, from 100 pictures, which pictures contain a cat, it really can't do it. And every 4-year-old can do it. So somewhere these technologies have to meet.
That was a very nice ending. Let's thank our speaker again.
Thank you.
Thank you for coming to this talk. Thank you to all of you online. And I think this concludes the talk.
Yeah, appreciate it. Thank you.
Thank you very much.